All right, welcome to this explainer. Today, we're diving into something completely mind-blowing, a hidden connection bridging two totally different worlds. On one hand, you've got the super precise, rigid math of computer science and telecommunications. You know, the stuff that makes the internet work. On the other hand, the chaotic, rapidly mutating biology of lethal viruses. By the end of our time today, you're going to see how the exact same math we use to send a text message without errors might actually be the key to designing the ultimate biological superdrug. It's wild stuff.
So what do telecommunications math and HIV drug resistance have in common? I know, it sounds like a trick question, right? Normally, we treat computer code and biological code as completely separate universes. But today, we're looking at this incredible overlap between MIT-level equations, literally designed for data transmission, and the survival mechanisms of one of the world's most adaptable viruses. To understand how we can possibly defeat a moving biological target, we've got to first understand how we send messages through the noise.
Act 1. The Mathematics of Communication.
Okay, let's rewind to 1948. A mathematician named Claude Shannon laid the groundwork for information theory. Basically, he was trying to figure out how to reliably send data from point A to point B. And that brings us to a massive fundamental problem, summed up perfectly by this concept from MIT. Linear codes are almost always examined with channels of additive noise. Now, what does that actually mean? Think of additive noise like static on a radio, or, if you remember them, a scratched CD. When you send a digital message through a cable or a wireless signal, this noise scrambles your data. It literally flips ones to zeros and zeros to ones, creating a bunch of errors. And if we don't fix those errors, the whole message is just lost.
So how do we decode the true message through all that static? Well, we use these mathematical tools called linear codes. What these codes do is add structured, predictable redundancy to our messages. It's basically a clever way of packing the data so tightly with mathematical rules that if a piece of it gets totally corrupted by noise, the system can just look at the surviving pieces, apply the rules, and automatically figure out what the missing piece was supposed to be. It's like a self-healing puzzle.
A perfect example of this in action is the Hamming code. Notice the overlapping circles representing data bits. The mathematical rule here is called a parity check. It's simple but brilliant. All 4 bits inside the same circle absolutely must sum up to 0. If static flips one of those bits, boom, the sum isn't 0 anymore, and the system instantly knows an error happened. But here's the real magic. By checking the overlapping sections, it can pinpoint exactly which bit was flipped and fix it on the fly. It is a completely self-correcting system.
Act 2. The Great Pivot.
Now hold on to that concept of parity checks because we are pivoting hard from digital channels straight into biological ones. Think about DNA and RNA as a literal genetic code, where biological mutations are the additive noise. To look at this through a biological lens, we really have to separate 2 terms, entropy and information. In Shannon's theory, entropy isn't just chaos. It's simply a measure of uncertainty or potential. Imagine a completely random string of DNA bases. That is high entropy. Information, on the other hand, is shared entropy. It's when that uncertainty actually means something specific relative to its environment. Like if a sequence of DNA perfectly matches a binding protein, well, that sequence isn't just random potential anymore. It holds vital, life-saving information for that organism.
Act 3. Reading the Biological Code.
If we treat biological mutations just like telecommunication noise, we start to see evolution acting as this massive, natural decoder. If a mutation, biological noise, hits a crucial part of a genetic sequence, the organism probably won't survive. But if it hits a non-essential part, the sequence keeps right on working. By mapping exactly where mutations can and cannot happen, we can read the biological code just like a computer scientist reads data. When we look at the entropy maps for tRNA in 2 completely different bacteria, E. coli and B. subtilis, the peaks represent high entropy, places where the genetic code is highly variable, or really noisy. The valleys? Those are low entropy, meaning those specific bases are strictly conserved because they hold crucial functional information. And here is the crazy part. Even though these 2 species are separated by 1.6 billion years of evolution, their maps look incredibly similar. A molecule's function strictly dictates its information profile, completely regardless of the species.
Which leads us to this amazing concept called epistatic correlations in RNA. Remember our Hamming code parity check a minute ago, where bits shared a mathematical rule to sum to 0? Well, RNA bases do the exact same thing. Certain bases in a genetic sequence literally share entropy. They act dependently on one another.
If one mutates, the other has to mutate in a complementary way just to keep the molecule's physical structure intact. Because of this functional association, we can actually use the math of shared entropy to predict the physical 3D structure of the molecule.
And this translates directly into physical reality. Look at the ladder-like stems in the secondary structure of a tRNA molecule, right where the bases pair up. Those pairs are physically bound together, meaning their biological fates are completely tied. Just like the bits in our telecommunications code, they share their entropy. By simply calculating the mutual entropy between all the possible pairs of bases in a sequence, we can figure out exactly which ones are physically touching each other.
The mutual entropy heat map proves it beautifully. The bright diagonal lines aren't just random data points. They are the exact mathematical correlations between bases. From these mathematical spikes alone, literally without ever putting the molecule under a microscope, we can identify the 4 physical stems of the tRNA molecule. We're reading the physical 3D shape of life by tracking shared entropy on a grid. How cool is that?
Act 4. Tracking HIV drug resistance.
Now, let's take these mathematical laws into a really high-stakes, real-world scenario, the battle against HIV-1. When you look at the entropy map of HIV-1 protease, which is a crucial enzyme for the virus, it is absolute chaos. All those spikes represent highly variable sites. The virus is actively using a massive rate of mutations, pure biological noise, as a survival mechanism. It intentionally maintains this polymorphic mixture of variations. That way, if we attack it with a standard drug, at least a few versions of the virus will have the exact right random mutation to survive and multiply. It's a brilliantly stubborn defense.
But math allows us to track its exact movements. If we measure the change in per-site entropy after a patient has been exposed to the drug saquinavir for 6 months, massive mathematical spikes suddenly emerge. These spikes identify the exact previously conserved regions of the virus that mutated just to resist the drug. We can mathematically pinpoint exactly where the virus altered its own code to survive our attack. Even wilder, we find that some of these resistance mutations happen in pairs. They act as a compensatory biological parity check, restoring the virus's function while completely evading the drug.
So how in the world do we defeat it? Well, the core problem is that a standard drug is a single static molecule. But HIV exists as a quasi-species, a chaotic polymorphic mixture. A single drug is always going to fail against a moving target. But information theory proposes a truly revolutionary solution: fight fire with fire. If the virus is an entropic mixture, the drug should be an entropic mixture of inhibitors too. We literally need a superdrug that mathematically co-varies with the virus. By matching the drug's entropy to the virus's entropy, we ensure that no matter how the virus mutates, a complementary inhibitor is already right there, perfectly tuned to bind to it and shut it down.
Which leaves us with this final, incredibly provocative thought to ponder. If a virus uses mathematical chaos as a shield, is the ultimate cure a drug that embraces that exact same chaos? By translating the rigid rules of telecommunications, things like parity checks, noise reduction, and entropy, into the fluid, messy world of biology, we aren't just reading the genetic code anymore. We're actually learning how to hack it. And that universal language of mathematics, it might just be the absolute key to finally curing the incurable. Thank you so much for joining me on this explainer, and keep questioning the hidden connections all around you.